Hello, my name is William Bains. I'm going to be talking about advanced glycation end products, cross-linking diseases in man, and possible treatments for them. It's pretty commonplace that we expect people to get stiffer, for the skin to get less elastic, for us to heal more slowly as we age. This has been observed for many thousands of years. What's relatively recently been discovered is why this happens and why this might be an approach to treating diseases of aging. So this presentation is about that. It's relatively non-technical, with one exception I'll come to. If you're interested in following up the really hardcore science, there's a bunch of references which are referred to down the bottom of the slides here, like this, and there'll be a list of references at the end. You can pause the video and have a look at them. We're going to cover protein cross-links, particularly advanced glycation end products, these things called AGEs, and their relationship to aging and disability. This isn't about genes, and about cells, so it's a bit unusual for a biology talk. However, it's important because we're made of proteins. Most of what we are is protein, and so the chemistry of those proteins is important. Proteins are very complicated chemicals, and so their chemistry is very complex. So the question is, when they do this chemistry, what happens next? Most of this talk is meant to be fairly accessible to a sort of undergraduate type audience. I will warn you, however, there is chemistry in here. So there'll be chemical structures. You don't have to understand what those chemical structures mean and the implications of the more detailed chemistry I'm talking about. But if you do, I think you get more out of it. This is my pitch for you to go out and become chemists. So, we learned at high school that living things are made of cells, and they are made of cells. This is the sort of diagram you get in school textbooks. Different sort of cells laid up. This might be, for example, the lining of your lung. Here's the epithelial cells on the top. This will be air above here, and various cells underneath. And this is right, we are, but we're not only made of cells. Lots of components of our bodies are made of things other than just cells. Some components are almost entirely made of non-cellular material. Things like hair, nails, don't have living cells in them. The lens of your eye is almost entirely non-cellular material called crystallines, and we'll come back to them in a minute. The enamel of your teeth is mostly not cells. Cartilage has a lot of non-cellular material, and so on all the way down through the components of your body. So cells are not the only story, and other materials outside the cells are important. But it's not just these tissues that are important from the point of view of not having cellular material, because around all our cells is something called the extracellular matrix. Great term. Extracellular matrix just means the stuff outside the cells, extracellular, that is a matrix, a support for cells to be embedded in. It's a sort of glue, if you like, but it's a lot more than just a glue. It's quite an important component of tissues that are connective tissues, hold bits of your body together. So things like skin, muscle, not just the muscle that bodybuilders build up, but the muscles that line your arteries, the muscles that line your guts and allow you to process food through them, tendons, bones, even fat has extracellular matrix holding it together in a particular structure. So this stuff is really important for the functioning components of the body. It's made of a wide variety of protein material. Collagen is one of the principal ones. Collagen is a very large protein molecule. If you take collagen out of something like tendon and boil it up so that its three-dimensional structure breaks down, you end up with gelatin. And gelatin is in fact denatured, broken down collagen. Inside your body it's not like that. It forms fibrils, long packets of protein molecules, sort of illustrated in this cartoon here by these lines. And this is pretty similar to what it actually looks like in life. Tissues such as tendons, which are meant to be relatively inelastic. If you try to pull a tendon, it doesn't stretch, have got a lot of collagen in. Other tissues that are more stretchy have other proteins in larger amounts, and particularly elastin, which is reflected in this cartoon as these sort of wiggly fibers here. Elastin, as the name suggests, is quite an elastic protein. If you pull it, it stretches. And then a whole load of other proteins, and this is a very complicated mixture of materials which I'm summarizing very simplistically almost here, that form a sort of glue, a ground base underneath these molecules, which interact with the cells, which interact with the elastin, with the collagen, with other proteins, and provide the chemistry and functionality necessary for the matrix. Embedded in all this in connective tissues are, of course, cells. And it's the cells that make these proteins. So these cells, and particularly the fibroblasts shown here, these will synthesize the proteins, export them from the cells, and then build up the extracellular matrix. I mentioned that different matrices have different effects and different chemistries. And this is really important because they are functional. They're not just an inert glue like the sort of carpet on the floor that cells sit on. They are highly functional. So this, for example, is a cross-section of the Achilles tendon. The blue colour, in this case, I think is elastin. The red colour is collagen. There's a lot of collagen, but a little bit of elastin. This is important because the Achilles tendon has to have a number of important functions. If any of you have torn your Achilles tendon, you'll know how important it is for running. It's because when you're running, when you put your foot down and stretch the tendon, that stores energy. And then when you release again, the tendon contracts, the muscles contract, that stored energy is released. That needs to be efficient. I don't know how many of you cyclists. I cycle around Cambridge. And those of you who are cyclists will know, that's the reason my bicycle's up here, that if you have a well pumped up tyre like this, cycling is very much easier than if you have flat tyres like this. A pumped up tyre is under tension. The rubber in the tyre is very efficient at storing energy. So when you compress the tyres, as the wheel runs along the road, it stores the energy, and when the wheel goes on, it releases that energy, so you don't put energy in to keep on squeezing the tyre backwards and forwards. When it's flat, that doesn't happen. And as you roll along the road, all the energy you're putting into the tyre to squish it and release it again is wasted. So it's very much harder to cycle with a flat tyre. Same thing with tendons. They have to be efficient. Skin, you don't want to have that property. You don't want skin to bounce backwards and forwards like a rubber band. If you bash your skin, you want it to absorb the energy and dissipate it. So different mechanical properties deriving from different chemistry of the extracellular matrix. Elasticity, strength, and so on. This matrix also has to interact with the cells. And there's some enormously complicated chemistry about how that happens, which I'm not going to go into at all. And it also needs to be able to be broken down and remodeled. So if you get injured, the cells need to be able to break down all the damaged extracellular matrix and rebuild up new ones. As you grow, as you develop, the extracellular matrix needs to be remodeled, re-engineered. All this relates to chemistry. So this extracellular matrix is a highly active and very carefully specified material and has to work right. It's not just proteins that are excreted by cells and then laid down and that's it. In order to provide this mechanical strength and also to provide some of the chemical functionality, the proteins are reactive with each other. They're cross-linked. So you've got a protein molecule here, a protein molecule here, and they are chemically linked together to form a bond between them. There are three types of cross-linking I'm going to mention in this talk. Two are formed by enzymes. One is formed by chemistry that happens without enzymes. The two enzymes are lysyl oxidase and transglutaminase. Lysyl oxidase is an enzyme family that cross-links collagen molecules. So I mentioned before, collagen molecules form these fibrils, long collagen molecules aligned together. They're then cross-linked, so they form an extremely tough structure. Transglutaminase, a family of enzymes that cross-link a much wider range of proteins. And these can be inside cells or outside cells. The ones we're interested in are the ones outside cells. I'll come on to advanced glycation end products in a minute. Those of you who want the chemistry, this is what lysyl oxidase does. It oxidizes lysine to this aldehyde, and then these can either condense to form a cross-link like this, which is then reduced to this stable structure, linking one protein here, another protein here through this bridge. Or two of these can condense to form an aldol condensation, which then goes through a number of other condensations and rearrangements to these 3 or 4 link cross bridges. Transglutaminase is a bit simpler. They take, again, a lysine and a glutamine and condense them together to expel ammonia and form this isopeptide bond here. This structure is called EGGL, and you'll see EGGL turning up in a couple of other slides. Those of you who think, oh, that's a bit much, the bottom line is that these crosslinks cross-link the matrix proteins and form a more stable and more rigid structure. So lysyl oxidase is important for cross-linking collagen fibers. Transglutaminase does a number of things. It builds the stratum corneum of the skin, a very, very top layer of the skin. The bit right on the outside has no cells in it. It's protein and lipid that's cross-linked to form an impermeable barrier between you and the outside world called the stratum corneum, and a lot of that cross-linking is done by a transglutaminase. Blood clotting, come on to this in a minute. It has roles in controlling inflammation, an involvement inside the cells on cell survival and so on. So a lot of different roles in the body for this enzyme transglutaminase. And advanced glycation end products, I'll come on to in a minute. Generally, the lysyl oxidase crosslinks do not increase with age, although they are increased in some diseases. The transglutaminase crosslinks do increase as we age, and also increase in some diseases and are known to be related to them. The advanced glycation end products definitely increase with age, and we're not quite sure what their role in disease is. So all these things are related to age or disease or both. Just to reiterate something, because those of you who want to dig into the science behind this and the research papers, you need to be aware of this. These crosslinks have different roles inside and outside the cell. Advanced glycation end products, lysine oxidases, transglutaminases all happen inside the cell, where they're related to the processes of the cell, control cell growth and so on, and outside the cell in the extracellular matrix. This talk is about the extracellular matrix part. That's not to say the other part's not important, but I'm going to focus on the extracellular matrix because I think that's a really interesting area and one that people generally do not think about when they're talking about these crosslinks. But do be aware, if you read a paper about protein crosslinks, are they talking about inside the cell, outside the cell? Do they just take the whole tissue and mush it up and analyze it? Sometimes people are not very specific about that, and you have to be careful. Okay, the one I've not been talking about, and the one I think is most interesting from a point of view of aging. Advanced glycation end products. These are the products of the chemical reaction of sugars with proteins. There are no enzymes involved. This is just cooking chemistry, chemicals together to make them react. And there are several steps in the process, and I want to go through this in a little bit of detail because when we come back to treating these cross-links and getting rid of them, this is going to be really important. So, sugars and proteins over the far side, the amino group here representing an amino group on proteins, this representing a sugar, react to form a shift space. This is a shift space structure, and then this can convert to what's called an Amadori product. This whole reaction is called the Maillard reaction, after the chemist who first described it in detail. This is a reversible process, so things can go backwards and forwards along this path, but if you've got enough sugar and enough protein, it will tend to all end up around these types of products. Then slowly, and with some oxidation, these products are converted to what's called advanced glycation end products. This is an irreversible process. At high temperatures, chemical reactions happen faster. At the temperatures of a typical sort of domestic oven, this sort of chemistry happens, well, this happens in seconds, this sort of happens in minutes, and this can happen in half an hour to an hour. And the brown colouring you see in roasted food, on potatoes and your chicken and so on, is actually these advanced glycation end products being built up. A lot of the taste and scent you get off food that's characteristic of food that's being cooked at fairly high temperature, such as baked bread, roasted meat, are these Amadori products. So this is actually quite important chemistry for the food industry. But from our point of view, it's important because it also happens in us. It doesn't happen nearly as fast in us as it happens in the oven, because we're much cooler. Chemical reactions tend to happen more slowly at lower temperatures. But it does happen in us. The sort of time scale in us is this happens in sort of minutes to hours to days. This conversion happens in months to years. And you can see the effects of this. You can actually physically see it. If you look at protein structure in a mammalian body that's usually completely colourless, like the lens of the eye. This is the lens of the eye from mice, but it could be from people. They'll look just the same. They're completely colourless, completely transparent, and of course they have to be. That's the whole point of the lens of the eye. It allows light into the eye so you can see. If you look at the lens of an old human being, and this is taken from a 67-year-old human, it's observably coloured. It's sort of going the same way as the roast potatoes down here. Now, it's taken nearly 70 years to do it rather than an hour because the temperature's lower. But these type of glycation end products are building up in that lens. And they build up in all your other tissues as well. It's just not quite as easy to see. A little bit more of the chemistry because this is important, and then we'll get onto the medicine. So here, over the far side, we've got our sugars reacting with proteins to form amidori products. These are then oxidized to form these key intermediates called dicarbonyls. These can then react with more proteins to form a complex mix of products. There's actually other ways you can do this too. Oxygen can oxidize the sugars directly to dicarbonyls, or lipids can be oxidized, and those can oxidize sugars to form dicarbonyls. But these are some pretty key intermediates in the middle. This mix of products is called advanced glycation end products. They're very stable. They're chemically stable, so they can sit in your body for decades and not break down chemically. Some of them actually fluorescent, so if you shine ultraviolet light on them, they will give out visible light in characteristic wavelengths. And some of them are coloured, so you get this sort of initially yellow and then light brown colour. Now, I'm a biochemist, so I'm happy with the idea of complex mix of products. If you talk to a chemist, they say complex mix of products is not a chemical description of something. It's just a description of soup, really. But that's about all I'm going to go into, except for a couple of exceptions on what those molecules are. And the reason is this. What forms in that mix depends on the sugars that are reacting, the amino acids that are reacting with them, and the sequence of the protein that's reacting, and the three-dimensional structure. So the same amino acid will have a different reaction in different proteins. It depends on the oxidizing agents that are subsequently oxidizing it. It depends on the time. It depends on the temperature. In us, the temperature is pretty uniform, but not completely uniform. The temperature of your skin, particularly the epidermis of your skin, is significantly lower than, say, the temperature of your liver. Here are some of the sugars that form Ages. This is just one protein structure to illustrate the complexity of that. If you're taking each of those and reacting with all the lysines in this, you're going to get a huge number of combinations. And you do get an enormously complex mixture. So the important one for us is which of these are most important in vivo, in a living organism. There are two categories of advanced glycation end products, of which I'm only really going to talk much about one. The non-crosslink advanced glycation end products are glycation end products which, if you've got a protein, are stuck on the outside. So they're modifications of the protein, but they don't link the protein with another protein. Ones people talk about are CML, carboxymethyl lysine, and we talk about as being common in man, which is used quite commonly as a marker for glycation in humans. And the methylglyoxyl hydroimidazolones. Even though I have problems with that, we call them MGHs. They have some very interesting biology, and my colleague and collaborator David Spiegel at Yale is discovering some fascinating things about how the body uses MGHs as part of its antioxidant defense. So they're formed randomly, they're formed by accident, but having been formed, the body says, I can use that. And this is an important point to bear in mind. We don't know the biology of what a lot of these things do. We're more interested in crosslinks. We're interested in glycation end products that have a protein here and a protein here and link the two. There's a whole zoo of them with names like MODIC and DODIC and Crossline and so on. The one we're interested in is glucosapane. This is the glucosapane structure. So here's one protein chain, here's another protein chain, and it forms this weird thing in the middle. This is the most common crosslink in extracellular matrix, certainly in skin, in man, by far, much more common than these other ones. And so research in our group, in our collaboration, is primarily looking at glucosylane and saying that is the one to focus on, both for finding out what it does and then seeing if we can get rid of it. An important aspect then is to say, okay, we get these things, how important are they? Well, this chemistry happens on a timescale of years to decades. We're used to, say, you know, cutting our hair every week or so, clipping our nails every week or so. Surely stuff doesn't hang around in us that long. Well, you can measure how long proteins hang around in you, and you do a very simple experiment. This is an example of that type of experiment that shows in mice proteins can hang around quite a long time. They did something really simple. They took mice and they started feeding them a diet dosed with carbon-13, which is an isotope of carbon. So you can trace where the food that that mouse is eating is ending up in that mouse. And then they looked at how much of that carbon-13 was present in the mouse with time. And then after a while they stopped feeding them that and switched them back to a normal diet. And they said, of that carbon-13 labeled food, you know, the food we'd best been feeding them, how long does it stay in the mouse? And obviously they labelled things that would tend to be incorporated into proteins. So they would look at how long the proteins stayed in the mouse. And this is two bits of data from this particular study. The top one is looking at liver. Here's the carbon-13 going into the liver. Here it's coming out again. And it goes in quite quickly and then it comes out quite quickly. Your liver gets turned over pretty quickly. Liver gets replaced, repaired, regenerated. Muscle, different story. Here's the carbon-13 going in, really quite slowly. Here's the carbon-13 going out. There's some of it, they stop feeding the mouse here, carbon-13 at about day 60. Some of it still here at day 160. Some of the proteins of this mouse muscle were lasting in the mouse at least 100 days. Now, lab mice only live for 2.5 years. This is a significant fraction of their lifespan. So we'd expect, if that was true of people, we could have proteins in us that have been in us for years, maybe decades. That's a long enough time to accumulate a lot of glycation end products. Now, of course, you'd reasonably say it'd be nice to do this experiment in people, but you can't ethically do that. Well, you can't, but actually somebody's done it anyway, because in the 1950s, the nuclear testing program dosed the atmosphere with a whole lot of carbon-14, which is the radioisotope of carbon. And that got into food, and then we ate it, and there was a huge surge in the amount of carbon-14 we all ate. Well, I ate. You're probably too young. So if you look at the carbon-14 in the air, this dark line here, it's stable until the early 50s, and then blasts like this injected a huge amount in the atmosphere, boom, and then it started to get washed out again. And you can use these curves to ask the question, how old are your proteins if you were born anywhere along this curve here? And it turns out that some of your proteins are very old indeed. The crystallins in the eye lens, remember the eye lens that was going yellowish up here? Well, those proteins have been in that eye lens, some of them, essentially since the eye lens was formed in early childhood. Your teeth enamel, the proteins in that are pretty much put down there, and then they're there to stay until the teeth fall out. Some other proteins are not quite so extreme, but they are there for years or decades. So there is plenty of time in man for these advanced glycation end products to accumulate to quite high levels. So this is an example of this. This is looking at proteins in lenses, in eye lens, and protein in skin. This is looking at carboxymethyl lysine, this CML stuff. This isn't a crosslink, but it's an advanced glycation end product. And you can see the amount going up with age. And we're gradually accumulating this stuff. This is a slightly more complicated looking picture, but it's exactly the same thing. This is looking at glucosylane, the crosslink, the one we're really interested in from cross-linking diseases. The blue guys here are in healthy people, and although they're scattered all over the place, the trend is clear that it increases with age. From age about 10 here in these studies up to this guy I think is 101. It's not an inevitable rule that it has to increase with age. So these two people here, age about 82, actually have less glucosapine in their skin, this is measuring skin, than this person here, aged about 16, 17. But the trend is there. What's really informative for talking about disease is the red ones. These are samples from people with diabetes. Now remember the glycation crosslinks, all the advanced glycation products are made reacting sugars with proteins. If you have more sugar, you'd expect more reaction, and that's what you find in almost any chemical reaction. Diabetics, of course, are people with abnormally high levels of sugar in their blood. If they're very well controlled, if they're treated with drugs and diet and exercise properly, the amount of extra sugar they have is quite small. If it's not so well controlled, it can be 2, 3, 4 times as much as their optimal level, the level in healthy people. And diabetics generally tend to have a higher level of crosslinks, this glucosapine, and some of them have enormously much higher levels. So, crosslinks accumulate with age. Glucosapine is the most common age crosslink, but is it important compared to the other ones? Remember I mentioned 2 other types of crosslink, the lysyl oxidase and the transglutaminase, both made by enzymes. People have looked at lysyl oxidase crosslinks and they find that these levels are somewhat higher than the lysyl oxidase crosslinks in the same tissues they're looking at. I don't have that data. I have some for transglutaminase crosslinks. And so here on the same scale is the amount of transglutaminase crosslinks in different tissues. And this is one of the frustrations of this. I'll come back to it in a minute. So up here we have transglutaminase crosslinks. And most of these measurements, these green guys down here, are down right at this low level here, under 500 picomoles per mg of protein, which suggests that it's certainly in older people, and most certainly in diabetics, glucosapine is more common than the transglutaminase crosslinks. What, you will reasonably say, about these green points up here? Well, those are samples taken from diseased tissue. So in some cases, when you've got particular diseases known to have a lot more transglutaminase in them, then you will get a lot more transglutaminase crosslinks. Then maybe glucosapine is a bit less important. And the frustration about this is that all these dots are measured in different tissues and different samples from all these dots. So it's very hard to say in a particular disease, a particular person, a particular tissue, this is what is most important on this, and this is what's less important in the crosslinks. It's something that does genuinely needs more research to sort out, and that's some research we're trying to do here at Cambridge. So, bottom line, advanced glycation end products accumulate with age, certainly in the skin, eye lens, kidney basement membrane. Glucosapine is the most common crosslink in those tissues. We don't know how much of the crosslinks accumulate in other tissues, particularly in blood vessels, that's important I'll come back to. It may be the most common crosslink in tissues of old people, but we don't know what the age relationship is for the other types of crosslink very reliably. So there's lots of unanswered questions around this. Given that uncertainty, we can ask, are crosslinks good or bad? And the answer is yes, they're good and bad. We can demonstrate how important they are by saying, can you do without them? So, some researchers have knocked out one of the transglutaminase genes, a transglutaminase that makes that stratum corneum on the skin. They've done some genetics on the mouse and they've just taken that gene out so the mouse cannot make transglutaminase. And the mice die almost as soon as they're born. They have no defense against losing water from their skin, so they just dehydrate. I mean, they dehydrate faster than the jellyfish in the sun. It demonstrates that that stratum corneum, that cross-linking, is absolutely crucial for life. Factor 13, one of the blood clotting factors, is a transglutaminase. Without it, you have serious coagulation disorders. Lysyl oxidase, people have also knocked that out of mice. They die very soon after birth from a variety of abnormalities. So yes, those crosslinks are absolutely crucial. You can't do without them. There's no way to knock out the advanced glycation end products. because they are not caused by a gene or an enzyme. They're chemistry, and you can't get rid of the proteins or get rid of the sugars from mammal. There may be ways of breaking them, and we'll come on to that at the end, but we won't know until we have broken them whether those are also functionally important or whether we can do them without them entirely. But it's certainly not the case that crosslinks are bad. However, in some contexts and some levels, crosslinks are associated with disease. Quite a wide range of disease. And so for the advanced glycation end products, a wide range of diseases are correlated with age, so that more crosslinks and more glycation end products are present in disease versus healthy people. And we saw an example of that with the diabetes. Functionally related to what glycation end products do and the crosslinks do, and I'll come on to an example of that in a minute. And they're also related to the body's reaction to advanced glycation end products. This isn't something I'm going to talk about anymore, but you can look up if you're interested. There are receptors in the body for advanced glycation end products, called receptors for advanced glycation end products, not very surprisingly, or RAGE. These are linked into the immune system. When the immune system sees a lot of advanced glycation end products, it tends to react. It causes inflammatory responses, which may be related to disease. And actually, if you measure the amount of advanced glycation end products in animals at a given age, how much advanced glycation end products they have in them predicts their life expectancy. Not reliably, but it gives a statistical indication. This works for a number of different animals. I don't know if anybody's done it in people. The problem with this is you don't know whether that's because the advanced glycation end products in those individuals are causing them to age, or whether it's simply a side effect. So, for example, if they have high sugar levels, they have a diabetes-like disease, they'll have more age, and they'll have high sugar. Well, which is causing them to die earlier? We don't know. I'm going to focus on crosslinks in the rest of this because I think this is the most interest to me, and I think it's something we can look to do something about. And talk a little bit more detail about a couple of diseases as examples of what we know about crosslinks and disease before starting to talk about how we might treat age-related crosslinks and the diseases that they may or may not cause. So, diabetes. We've talked about diabetes before. More advanced glycation end products are clearly seen in diabetics. And furthermore, the more advanced glycation end product you have, the more likely you are to have complications of diabetes. So, nowadays diabetes in itself, the inability to make insulin in itself, is not a very serious disease because it can be treated extremely effectively with drugs, including with insulin. So diabetics can live a long, long time. 100 years ago they couldn't. They died quite quickly. If they got onset in their teens, they would expect to die in their 20s. Now, diabetics can live a long time. What kills diabetics today are complications of their disease, and particularly heart disease, cardiovascular disease, and kidney disease. And people have looked at the chances of getting several different sorts of end-stage renal disease, kidney failure, and diabetes, and they found the more advanced glycation end products you have, the more likely a diabetic is to have serious complications from the kidneys. This may be related to cross-linking of the membranes in the kidneys, that extracellular matrix. The kidney cells that are filtering unwanted materials out your blood all the time, concentrating them into urine, they're sitting on extracellular matrix, and if that isn't working properly, then those cells could well be having a problem. In fact, this correlation between glycation end products and kidney problems, other problems in diabetes, is now so well established that fluorescence I mentioned, the fact that these advanced glycation end products will glow when you shine UV light on them, is starting to be used as a diagnostic tool to follow how well diabetics are being controlled and the chances of them getting end-stage renal disease, cardiovascular disease as a result of a complication of their underlying diabetes. The problem for us is that this graph here, which you've seen before, is a great correlation. But does it mean that the glycation end products are causing the disease, causing the secondary effects, the complications of the disease, or is it just a flag, a marker? So we could imagine two arguments, okay? Diabetes means worse sugar control. Worse sugar control may in itself cause physiological damage, which means more complications. Worse sugar control certainly results in more advanced glycation end products. Does that cause physiological damage and has more complications, or is this branch actually irrelevant? Well, that's not really known because you can't separate these two out. You can't say, treat diabetics with things that will get rid of the ages and see what happens. And we come back to this problem of causality. What causes what? What causes what in biology is an extremely difficult thing to take apart because biological organisms are so complicated. A second type of disability are the things we associate with being old. Feeling old, looking old. Feeling old, stiffness, loss of elasticity in the tendons. Young children can fall off chairs and laugh. If an 80-year-old falls off a chair, it's something that can be a very serious injury to them. This is substantially because of loss of elasticity, of resilience in the tendons, in the bones, and the muscles. And you would expect that if you cross-linked all those proteins, they become more and more like a rigid bit of plastic and less like an elastic band able to bounce. It's not quite that simple because a lot of that loss of function is due to inflammatory disease, either rheumatoid arthritis or osteoarthritis, or the sort of low level of inflammation that gradually builds up with age. And how much of the cross-linking is due to the inflammatory disease is due to cross-linking, and so on, is not known. It's very hard to dissect out. And there's also a loss of repair capacity with age. So if you tear a muscle, tear a tendon when you're older, it heals very much more slowly. Damage anything when you're older, it heals more slowly. And that means that those little tears and pulls in muscles as you run upstairs just a bit too fast, when you're 17, within an hour or two you feel fine. When you're 70, it takes days or weeks before you feel fine again, if you ever do. This is related to stem cells. We're going to talk about stem cells in a minute. One of the most obvious changes with aging is the physical appearance of aging, particularly loss of skin elasticity and wrinkling. There's a multi-billion-dollar industry dedicated to covering up the effects of aging as it relates to wrinkles. Then the amount of anti-wrinkle cream sold is absolutely massive. Some of this is because the skin is less elastic. So when you smile and when you frown, the skin gets stretched and it doesn't quite snap back. And so you end up with a wrinkly skin, as opposed to a 3-year-old smiles and frowns, and when they stop smiling and frowning, the skin goes flat and smooth as if they'd never had the expression at all. And this you'd expect to be a result of loss of mechanical elasticity in the skin, and indeed you find that. So this is one measure of elasticity in the skin. This is just a particular measure for a particular machine. There are lots of ways of measuring elasticity, and this is correlating this with age. And again, you see a downward trend, loss of elasticity with age. As with the glucosapane crosslinks, it's not inevitable. This person here, age of about 56, has skin that's at least as elastic, as bouncy, as this person here, age about 26. Because other factors are involved. Sun damage, smoking, genetics, these all influence how much you lose elasticity in your skin. And also your skin gets thinner with age, so it shows the wrinkles more. And this, again, is related to stem cells. But it's reasonable to suppose that both of these types of categories of aging effects are related, at least in part, to increasing cross-linking of the proteins in the extracellular matrix and loss of elasticity as a result. One of the best established disease implications of loss of elasticity is in the heart, in the cardiovascular system. So, here we've got the heart. The left ventricle over here drives the blood around the body. It compresses, squeezes the blood out, this volume here, and into what's called the conduit arteries, the major arteries like your aorta, carrying the blood down into your body where it spreads out into the different tissues. This is not just a pipe. This is a complicated, muscly organ that's got muscle on the outside, cells lining it, and then obviously blood in the middle. And when the pressure goes up on that organ, it relaxes and expands, so there's more space inside it. And then when the pressure drops, when the heart stops contracting, the muscles squeeze to reduce the volume again and bring it back to its original size. So this means as the heart contracts, the volume of the blood vessels expands and the resistance for the heart pushing it goes down because they've got more space to push blood into. When the heart's contraction is nearing its end, the blood vessels then help it by contracting themselves and pushing the blood on. And the net effect is if you look at the pressure of the blood in these major blood vessels, you get a curve. So here's time along here. This is measuring from the start of this surge of blood through the blood vessels to out to about a second. And you get this, if you didn't have this elastic effect, you get this sort of red line here. The pressure would go up and then it would go down. This is the heart squeezing, squeezing, and then it stops squeezing. Because the blood vessels respond to this contraction, this peak pressure here is reduced and the overall duration of this pulse of high pressure is stretched out. So the changes in pressure are not so extreme. And this is really important. If you reduce this elasticity, make these vessels stiffer so they can't expand, then you get increased pressure here. You get a reduced pulse, you get greater extremes of pressure. And this can cause damage. It causes damage to the heart. The heart says, I have to work harder, I have to put more force in, and so it tends to make more muscle, get what's called cardiac hypertrophy, which is actually a bad thing above a certain point. It damages the inside of the blood vessels, and it results in a range of different damage which I'll come on to. But this has a very, very simple measure, and this is why people know a lot about this. Because this peak here, when doctors measure your blood pressure, you go in and they say, oh, your blood pressure is, 155 over 80. Mr. Bains, you really, really ought to do something about that. 155 is this up here. If I could reduce that to 130 down there, I'd be a healthier person. And that's a reflection, not of the fact that a number is a good thing and another number is not a good thing, but because it's a reflection that those sort of high pressures and rapid changes of pressures are bad things. A lot of the hypertension that occurs with old age is not because of eating too much fat or eating too much salt. It's because of this stiffening effect. And the reason people say you should cut down on fat, cut down on salt, and get exercise is to try to counteract this as you get older. So the blood vessels do get stiffer with age. This might be because of advanced glycation end products. It certainly is due to the transglutaminases. Remember I said that the crosslinks were called EGGLs. Well, those have been measured. That might be related to inflammation. Loss of elasticity leads to hypertension, can lead to cardiac hypertrophy, certainly leads to damage of the arteries, which leads to atherosclerosis, which leads to more stiffening and more inflammation, and you get into this vicious cycle. You end up with arteries that look like this. This is a cross-section to a really sick artery. This whole inner layer here shouldn't be here. This mass here certainly shouldn't be here. This is an artery that's been taken out of somebody's heart undergoing bypass surgery. If your arteries end up looking like that, you've got problems. This stiffness can also damage the endothelial cells that line the artery and are really important as signaling to the whole blood system how much pressure is in the artery, whether the blood system, whether the arteries need to relax or contract. Damage them, then you get again into a vicious cycle of loss of control of blood pressure and has more damage to the artery, and they can't repair themselves, so you end up with atherosclerosis and things. So, stiffness, really bad thing. And I mentioned, go into this in several slides because we're going to come on to how people have looked at this as a potential outcome to be treated by getting rid of advanced glycation end products in a minute. I would emphasize, again, that some crosslinks are essential for the strength of the arterial wall. If you didn't have things like lysyl oxidase, crosslinks in this arterial wall here, the arteries would burst. So it's the balance that's important. I mentioned stem cells, and the last thing I'm going to get onto about diseases is stem cells before we talk about potential treatments at the end. So stem cells are cells that can replicate themselves to make more stem cells and make other types of cells. Diagram right at the start of the talk looked like this. There were all sorts of different cells in it. Some of these cells can divide to form the same sort of cell. So these are meant to be smooth muscle cells, and a smooth muscle cell can divide to make two smooth muscle cells. However, it can't divide to make a nerve cell and a smooth muscle cell. Nerve cells here quite often can't divide at all. So if you wound this, if you brush up against a wall or a thorn and tear a hole in this, if this was your skin, how do you get this nerve cells, smooth muscle cells back? Well, for that you call on stem cells. And stem cells can grow to maintain a small number of stem cells, but when needed, can also grow out and then turn into other cell types. The whole field of stem cells is an enormous area of biological research at the moment, both in basic biology and in applied biology and regenerative medicine. And I'm sure there'll be another talk in this series on stem cells. Stem cells can be specific for a particular cell type or tissue type, or they can be much more general, what's called multipotent stem cells or pluripotent stem cells that can generate lots of different cells. But a common feature of all of them is that they're not just distributed at random around your body. They need a specific type of environment to grow and to remain as stem cells and to turn into their correct target cells. This environment is other cells around them and their extracellular matrix. And together, this is what's called a stem cell niche. You have lots and lots of stem cell niches around your body, little repositories of stem cells where they're kept happy and kept as healthy stem cells, able to grow and able to repair you and rebuild parts of you as necessary by the extracellular matrix and by the cells around them and the interaction of all that chemistry. This is a very specific chemical environment, and so you would expect that if you damage that environment, changed it in some way, you damage the stem cell's ability to grow or to continue to survive as stem cells, and indeed we find that's the case. So, if you take old stem cells from an old animal and you get other cells to make extracellular matrix from that old animal, it makes old extracellular matrix, you put the stem cells on it, the stem cells, they're okay, but they're not great, they're not very happy. If you get cells from a young animal to make young extracellular matrix, and then you take old stem cells and put it out young extracellular matrix, it's as if they've been rejuvenated to get a new lease of life. They're not completely rejuvenated back to the young stage, but they feel a lot healthier. You could do something even bigger. The regenerative medicine people will tell a lot more about this. But in summary, you can take an organ, like a kidney, and you can wash all the cells out of it. And what you're left with is a kidney-shaped piece of extracellular matrix. You can then add cells back to that, and they'll find their way through the extracellular matrix to where they ought to be and rebuild you a functioning kidney. At least in principle. It turns out to be rather difficult to do. It doesn't work 100% yet. And of course, you started out with a kidney and you end up with a kidney, so you haven't built a new kidney for anybody. However, the principle is you can build new organs by having the right extracellular matrix around them. Extracellular matrix you can get from old organs. If you do this with a young kidney extracellular matrix, it works much better than with old extracellular kidney matrix. Now, extracellular matrix from kidneys. Don't forget that these extracellular matrices have no cells in. This has nothing to do with cells getting old, DNA getting damaged. This is just the extracellular matrix. But young is better than old. Third bit I've just got down here, if you take advanced glycation end products from proteins from extracellular matrix and just feed them to cells in the proverbial petri dish, those cells tend to grow more slowly, divide more slowly, move around the dish more slowly. Is generally bad for them. So, yes, advanced glycation end product damage the extracellular matrix, certainly could cause damage to the stem cell niches. There are other factors involved in aging that also affect stem cells. immune system effects, hormone effects and so on. This isn't the only reason that stem cells of someone at 70 don't seem to be as energetic as the stem cells of someone who's 7, but it may well be a reason. The importance of thinking about stem cells in the extracellular matrix is that this is relevant to all diseases and disabilities of aging. It's not just a heart disease or brain disease. Regenerating any part of our cells relies on stem cells. Repair, maintenance relies on stem cells. So fewer active, healthy stem cells means lack of tissue repair and lack of disease damage from artery disease, inflammation, and also a loss of tissue, because we're growing and repairing and rebuilding all the time. And if the rate of losing cells is steady, but the rate of replacing them starts to drop, of course we'll lose tissue. And that's why the skin thins, that's why the very old people suffer what's called sarcopenia, which means losing muscle mass, basically. Brain shrinkage in old age, all these things are because the stem cells have said, hey, you know, I'm just not really up to it today. I'm going to stay in bed rather than building some more tissue. We don't know how much of this is due to crosslinks and how much is due with glycation end products. So, bottom line on glycation end products. Clearly increase with age, maybe a major crosslink in the older extracellular matrix. Good reason to suppose they affect health in direct and indirect ways, but we don't know the real contribution they make. They may be really important, we don't know. And the only way to find out is to try to get rid of some and see what happens. This means we need what's called an Agebreaker drug. A drug that will crack open ages and get rid of them. And that's what I'm going to finish up on. How do you get rid of crosslinks? Well, there are really 3 ways that we can think about. We can stop them forming in the first place. In the case of advanced glycation end products, we can't block enzymes because they're not made by enzymes. In principle, we could block the chemistry that happens. We could enhance mechanisms that break them down. This is internal enzyme mechanisms in the body that break down crosslinks, not relevant to advanced glycation end products, because as far as we know, there are no enzymes in our bodies that break down glycation end products. There are some enzymes that break down the first products in that chain of reaction that led to them, called fructosamine 3 kinases. But they're not relevant to ages for 2 reasons. Firstly, because they don't work on ages, they're only on the first shift-space and Amadori products, and secondly, because they're inside cells, not outside. So we have to add something to break them down, and that something could be an enzyme or could be a chemical. And these are maybes. Yes, in principle, we could do that. But finding something like that is really tough. So, stopping things forming, there are several good reviews on this, which I mentioned in the reference list, so I'm not going to talk about this a lot. In principle, you can eat something or inject something that'll block any of those reaction intermediates from the sugars and proteins all the way through to advanced glycation end products. And hence, if you stop ages forming, then they won't accumulate. The intermediate that people focus on are those dicarbonyls. Remember I mentioned those as being a key thing in the middle of the diagram. Those are reactive compounds, and so you can put a chemical in that will block them, then they won't go on to form ages. A couple of things people have talked about, aminoguanidine here, maybe metformin, which is marketed as a diabetic drug called glucophage. It's not really clear whether this does anything for age at all, or whether what it actually does is treat diabetes, which of course is associated with more ages. They sort of work, but the problem is that once the ages are formed, then they're there. So all you're doing is reducing the rate at which they're formed. There is reason to suppose that might be useful, but it's not the ultimate goal. The ultimate goal is to find an age breaker, something that will actually cut crosslinks. The only molecule found so far that's got potential for doing that is this thing. It's called Agebreaker. It's also sometimes mentioned in the literature on the web as ALT711. This is meant to actually cut dicarbonyls, chop them. the chemistry around this is quite controversial as to what this is really doing, but it certainly does something in animal models. And it does something in animal models of hypertension. It reduces age-associated hypertension. It reduces a number of other measures of cross-linking, such as tendon elasticity. So it does something really cool, and it does something sufficiently cool for people to be willing to say, let's try it in human beings. So Agebreaker was put into human beings with age-associated hypertension, 23 patients, 16 weeks, and no change in blood pressure. A small change in some aspects of heart function. One of the problems with doing this is that there's enormous variability in blood pressure and enormous variability in crosslinks. You add those two variabilities together and you've got dots all over the place. This is just an example. This is the systolic blood pressure, the peak on that chart, of a whole population of people between the ages of about 45 and up to 100. And if this was a nice clean line here, we could say, ha-ha, we've taken the blood pressure from there down to there. That proves something. But people have blood pressure all over the place. So it's really hard to prove your drug's having an effect. So maybe the problem is we just haven't tried enough people. Actually, it looks like the problem is it's not having the effect we want, because there's another trial on at the moment with a much larger number of patients being treated for a much longer time. The formal results aren't out yet. The initial results do suggest that Agebreaker is not doing anything. So yes, it cleaves dicarbonyls, but whether that does you any good is not yet known. Options for the future then, if this isn't the right molecule, what might be? Well, you could look for an enzyme that chops this structure up. The problem with that is this structure is sort of buried in between proteins. It's going to be quite hard to find an enzyme that can really get at that. This is what's called the in vivo bioremediation. Bioremediation is clearing up environmental waste using microorganisms. And, well, if this is just waste accumulated inside our bodies, why don't we use the same logic? And that's the logic of this. There are groups looking for this at the moment, for an enzyme that will chew this up. Not yet found anything useful. It's likely to come from a fungus or a bacterium or something, but that's no problem. If it can be made and cleaned up and purified and used as a drug, then we don't care where it comes from. The other route is to find a chemical, a specific chemical that will react with this structure here and break it. That doesn't have a problem how it's going to get to the crosslinks. The problem with that is what on earth are you going to find? You need to find something that will discriminate this structure from all the other chemical structures that are vaguely similar in the body. I mean, we know chemicals that will break this. Boiling this up in concentrated acid will break it very, very efficiently. But boiling people up in concentrated acid obviously is not a useful drug treatment. The problem is finding something that's specific. So, there are routes forward, but none of them have really got very far. And this is a real chicken and egg. Until you can demonstrate that an Agebreaker is valuable, you won't get major investment from pharmaceutical companies in developing a drug. Until you've got an Agebreaker, you can't test it out to say, yes, breaking ages really is valuable in that disease. So there's a sort of circularity and chicken and egg problem. And breaking these things is a tough nut to crack. These are difficult problems to do. I think the reason for being optimistic about trying to find an Agebreaker is it doesn't have to be 100%. Let's go back to this graph for one last time. Even people down here in their teens and 20s still have a significant amount of glucosapine in this case in their skin. We don't have to get rid of all the crosslinks. All we have to do is tip the balance a little bit to turn somebody around here at 60, 70, 80 back to the level they have here. And our proteins are being turned over, apart from one or two things like the lens and the enamel of the teeth. So we don't have to solve the whole problem. Just a little bit of clean up, a little bit of damage repair might be enough to do some real good. So that's what I hope will happen. Those of you who want to follow this up, I hope you can see this on the slides. Pause the presentation here. You can copy down the references. I'd just like to finish up by saying some thanks. The Agebreakers project is the focus of the Cambridge research group here. This is funded by the SENS Foundation. The SENS Foundation is a Californian-based charitable foundation supporting research into regenerative medicines and regenerative biotechnology approaches for treating the diseases and disabilities of old age. They have realized that the problem with developing such technologies is not that in theory you can't develop them, but that nobody really believes it's worthwhile until somebody demonstrates that there is potential there. And so they've funded a lot of really speculative programs to try to demonstrate that regenerative biotechnology and regenerative medicine can treat underlying damage related to age and hence related to the diseases and disabilities of age. I think that's a fantastic goal, and there's far too little investment in biomedical research that's not looking at the standard mainstream treatments, standard mainstream technologies. That's trying to look to the real future. And if there's anything you do to help the SENS Foundation, please hit their website and talk to them. They're really worth supporting. I'm obviously enormously biased. I've worked with the founder of the SENS Foundation, Dr. Aubrey de Grey, for about 15 years now, and they're funding the work that's being done here in Cambridge and I'm working with. So yes, I'm biased. I'm happy to work with them because I think it's a great cause. I'd also like to thank David Spiegel, who I mentioned at Yale, who's done some chemistry on this, Shay Soka at Wake Forest, who's working on some of the extracellular matrix aspects of regenerative medicine, and Rhiannon Granger, who's working upstairs at this very moment trying to discover Agebreakers, and to you, ladies and gentlemen, for your time and attention.